My name is Dr. Alma Solis of the Systematic Entomology Laboratory. I work at the National Museum of Natural History at the Smithsonian Institution, Washington, D.C. This talk is about the larvae of Pyraloidea, or snout moths. These are 4 pictures of larvae of the Pyraloidea. As you can see, 3 of them are very brightly colored, and this comes from the plants that they feed on. The Pyraloidea larval habitat diversity is high. The larvae have developed a broad array of concealed feeding strategies, folding, rolling, webbing, or tying of leaves, making tunnels or tubes of silk and/or frass. They mine leaves and bore into stems, roots, buds, and fruits. Larvae have diverse habits. They consume dried or decaying plant or animal matter, wax in bee and wasp nests, and living plants. Some are known to be inquilines in ant nests, predators of scale insects, and aquatic scavengers in flowing water. Many pyraloids are pests to a wide variety of crops and stored products and rank as among the most destructive pests to gramineous crops such as corn, sugarcane, and rice. A number of species have been investigated for biological control of noxious weeds, such as the cactus moth, but it has become an invasive noxious pest itself, and also in the biological control of aquatic weeds. Many pyraloid larvae have concealed feeding strategies. They fold leaves down, they're in seeds. This is a picture of a caterpillar building a shelter from a leaf. And basically, they lay silk down back and forth, and then they finally put one extra strong silk, and the leaf closes in a millisecond. They live and work in fecal matter, and a lot of people know these moths as the caterpillars that live in feces. They don't seem to be threatened by bacteria or anything that's growing on the fecal matter. Pyraloidea larvae as pests. This is how I see them. You remember the first slide where they're brightly colored. By the time I get them, where they've been boiled and put in alcohol, you don't see very much. But what you do see are sedal patterns. And so many of the identifications and the knowledge about what the species looks like is based on the larval ketotaxy. This is a musotomine caterpillar from Southeast Asia. How to distinguish Pyraloidea larvae from most primitive moths. Pyraloids, noctuids, and other derived lepidopteran taxa have 2 setae in the presporacular group of the prothorax. But many primitive lepidopteran taxa, for example, Gulecchioids and Tortricids, have 3 setae in the presporacular group of the prothorax, which can be seen here. Exception is how to distinguish Pyraloidea from Carpocinidae, also with 2 setae on the presporacular group of the prothorax. Pyraloids have 3 subventral setae on the abdominal segments of the prolags, and you can see them here. Carpocinids usually have 4 subventral setae associated with the prolags, but the number may vary from segment to segment in the abdomen. How to distinguish Pyraloidea from Noctuoidea with 2 setae on the presporacular group of the prothorax. Pyraloids have the crochets of the prolags in a complete circle or penellipse. Exceptions include the aquatic and the semi-aquatic immatures. So here you can see the circle, the complete circle, and the penellipse. Pyraloids can be distinguished from noctuoids because they have the crochets of the prolags arranged in a mesoseries, shown here. The Pyraloidea has 2 families. The Pyralidae specifically can be identified by the pinaculum ring at the base of SD1 on A8, seen here. They also can have 3 setae in the L group on A9. And in the bottom left, you see exception, Etaella cincinella. Etaella cincinella may lack these characters or have any combination of these characters, or reduced setae. Pyralidae with unusual biologies or morphological structures. In the Phycetines, many are seed feeders and therefore known worldwide as stored product pests, but a few species are known to be predators of scale insects. And what you see here are scales and the webbing of a Phycetine caterpillar. And they've been munching on the scales. This is why this looks like a mess. Phycetinae can be identified by the pinaculum ring at the base of SD1 on the mesothorax. Glirrini, many have symbiotic relationships with bee and wasp nests, including the well-known wax bee moth, Galleria melonella. What you see here are pupae of Galleria melonella. It can be identified by the pinaculum ring at the base of SD1 on abdominal segment 1. Chrysogyny, the pinaculum ring is at the base of SD1 on the metathorax. Cranbourne, how to distinguish Cranbourne larvae from Pyraloidea larvae. One, sometimes two setae in the L group on A9. Pyraloidea has 3 L setae on A9. You can see it here. This can sometimes be 1, but most of the time it is 2. There's no sclerotized ring around the base of seta SD1 on A8 that is present in Pyraloidea, although pinacula may be present. Cranbourne with unusual biologies or morphological structures. The Acentropini, many are aquatic, that is, their bodies are modified to live and breathe underwater or in areas of high humidity. These are often sent to me with aquatic plants. They have very many larval morphological structures associated with breathing underwater. On the left, you can see a tracheal gill for oxygen exchange. You can see the crochets of the prolegs in an ellipse, and they have rugose integument for plastron formation and cuticular respiration. Ctenobiini, many are stem borers near water and have a ventral prothoracic sac near with function unknown. Musotomini, many are known to feed on ferns and have been tested as biological control agents of noxious and or invasive ferns. This was also the first time a leaf miner was authenticated in the Pyraloidea. Cranbourne, it has over 1,800 species. They have 2 general habits. They are root feeders or stem borers. Root feeders cause damage to turf and sod, but also corn and other field crops. Stem borers are some of the most destructive pests of economically important grasses, such as corn, sugarcane, and rice. They also bore into sedges and rushes. Cranbourne or sodweb worms pass the day in silk-lined tunnels in the ground and emerge at night to feed on sod grasses. The giant Myolobia, Smyrintha, bores in the stems of bamboo. How to identify Cranbourne larvae? They have a single transverse plate posterior to the dorsal pinacula of the mesothorax, which you can see here. And they have crochets on the prolegs are in a complete circle. How to distinguish Diatria and chylo from Eurema larvae? Diatria and chylo have 2 subventral setae on the meso and metathorax. Eurema only has 1 seta. There are no pigmented areas around the lateral setae on proleg-bearing segments of Diatria and chylo, and Eurema has these pigmented. How to distinguish Diatria from chylo larvae? Chylo larvae have 1 mid-dorsal stripe and 2 lateral stripes. Diatria larvae do not have a mid-dorsal stripe and may or may not have lateral stripes. How to distinguish Diatria saccharalis and Diatria lineolata larvae? Again, most common species. The innermost seta of the anal proleg, the paraprox seta, as seen from the ventral view, is shorter than the next lateral seta in D. lineolata, and it is nearly the same size as the next lateral seta in D. saccharalis. How to distinguish commonly intercepted Diatria larvae? There are many species of Diatria in the Western Hemisphere, but very few larvae can actually be identified to the species level. If on A3 through 6, pinacula of seta SD1 extends to level of the middle of the loral or lower of the spiracle in front of the spiracle, and seta SD2 is at the bottom of the spiracle or lower, this is probably Diatria grandiosella. Its distribution is southwestern US and Mexico, and its hosts include corn, sorghum, and sugarcane. It's interesting to note that Diatria lineolata looks like Diatria grandiosella, but the pinacula are less sclerotized, and this species occurs in Central to South America and the Caribbean, and its host is corn. On A3 through 6, pinacula on seta SD1 barely extends below the level of the top of the spiracle in front of the spiracle, and SD2 is at the level of the middle of the spiracle. Then it can be 1 or 2 species. From North and South America and Caribbean, the hosts, sugarcane, corn, rice, or sorghum, it is probably Diatria saccharelis. If it is from northwestern Mexico, from Sinaloa, Nayarit, possibly Jalisco and Colima, and hosts sugarcane only, it is probably Diatria considerata. Note, Diatria magnificatella looks like Diatria considerata, but occurs in eastern Mexico states. Its hosts are diverse, but Box, who did much of the seminal work on Diatria larvae, admits that misidentifications are a problem in this group historically. And these are selected and/or cited references.